Hi, I'm Steve Pelech, the President and Chief Scientific Officer of Kinexus Bioinformatics Corporation. Thank you for tuning in to my presentation, and I also like to thank the organizers for allowing me the opportunity to tell you a little bit about what's going on here at Kinexus Bioinformatics Corporation, specifically with respect to our bioinformatics programs. This topic of the conversation today is about the Oncoproteome and its study. And if we look at cancer research today, in terms of the prevalence of the disease, and this is data from the American Association for Cancer Research, we see that the most common forms of cancer of the breast and the prostate. And other tumors also occur. They're solid tumors. And then the leukemias are not quite as common, but obviously do represent a significant portion. If we look at the percentage with respect to risks, we find that 1 in 6 men will get prostate cancer and about 1 in 8 females will get breast cancer. The other types of cancer occur less frequently, but in general, if you're a male, your chances of getting cancer is about 1 in 2, and as a female, 1 in 3. Now, if we look at the life expectancy with these different forms of cancer, what we see is that over the last 50 years, in terms of cancer at all sites, there's only been a marginal improvement of about 15% above what we saw in the survival rates from about 50 years ago, 15 to 20%. And the greatest success has been in things like breast cancer and prostate cancer, and these reflect early diagnosis. But in the case of the more other solid type tumors, if we look at the pancreatic cancers, lung cancers, ovarian cancer, really, despite the fact that there's been this war on cancer during this period of time, after more than 50 years of intense research funding, the NIH was set up under the Johnson administration about 50 years ago in the early 60s. Really, the survival rates have only increased by about 20%. And this is problematic because the incidence of cancer has actually increased. And so still today, about 1/3 of all patients diagnosed with cancer die within 5 years. When we look to see where the success has been, obviously things like prostate cancer and breast cancer and some forms of leukemias and lymphomas, this is where a lot of the research funding has been, largely because the lymphomas and leukemias are easy to study as single cell type cancers. With respect to the others, it's really early diagnosis that's helped a lot. Now the problem is that despite the fact that we've sequenced the genome about 10 years ago, we still don't actually know what about 40% of the genes do. And it's an estimated something between 21 to 22,000 genes that are coding for proteins in the human genome. And of these, about 3,000 are linked to the development of cancer, one way or the other. So the real problem is that every single human tumor that's primary may in fact be unique in its etiology. And every tumor may be heterogeneous within its population of cells in the types of mutations that it features. So the complexity of the human genome and the proteome is a major problem with respect to a barrier for improving diagnoses and treatments of cancer, simply because of the complexity of the disease. Now, my presentation is going to address a few different features. I'm going to summarize the efforts of Kinexus to create an open access bioinformatics database with websites for the study of cell signaling systems, both under normal and neoplastic conditions. And the opportunity here is that this can help facilitate research in this field. I'm also going to summarize some of the results of queries of these databases with respect to both gene mutation and diverse human cancers. I'm going to introduce the concept of tumor-requiring proteins. We've heard of oncogenes and we've heard of tumor suppressor genes. And I think there's a third category of genes that's actually even more interesting for their potential for therapeutic development. And finally, I'm going to especially focus on protein kinases as diagnostic markers and therapeutic drug targets. So if we look at today, a lot of interest is in the methylation of genes and with respect to epigenomic studies. And of course, the methylation genes can turn them off. But if they're activated, we get the production of mRNAs, and these in turn can be regulated by the production of about 1,000 different mRNAs encoded by the human genome. And of course, these facilitate the degradation of the mRNAs before they're translated into proteins. If they are successfully translated into proteins, most of these proteins may in fact be inactive. They can be rapidly degraded by proteases, but to be fully active, most proteins undergo covalent modification. More than 100 types of covalent modification are known, and the premier form of modification is protein phosphorylation catalyzed by protein kinases. And about 3 times as often as inactivation is activation of proteins by their phosphorylation, where functional information is known. And so when we look at a phenotype like cancer, it's really a reflection of the amount of active protein that's available that gives rise to the phenotype. And these proteins serve really as the main targets for drug development. Protein kinases currently represent about 1/3 of all R&D activity in the pharmaceutical industry. So this is really where a lot of focus at Connexus has been over the last 15 years. So again, the human proteome has about 21,500 proteins roughly. And out of that, a portion called the kinome contains about 536 to 538 protein kinase genes. And these undergo alternative splicing, which can give rise to other isoforms. These kinases catalyze the phosphorylation of most of the proteins encoded by the human genome, in fact. And over 200,000 phosphorylation sites have been demonstrated experimentally by mass spectrometry. At Connexus, we've used advanced algorithms with training from these 200,000 phosphorylation sites to predict another 1 million, a total of about 1 million human phosphorylation sites, an additional 800,000 roughly. And this number is in fact quite realistic. We know about 4% of the phosphorylation sites identified by mass spectrometry in random studies where there isn't enrichment for phosphotyrosine. There's about 40,000 sites that are known, and 4% of the sites. So you can kind of back calculate that, in fact, that does support a number close to 1 million human phosphorylation sites. If 1 in 25 are on tyrosine and there's 40,000 of these sites, then 40,000 times 25 gives you close to 1 million. Now these sites are dephosphorylated by a phosphatome, which consists of at least 156 genes of code protein phosphatases for the reversible reaction. Now we've created the SigNET website a few years ago and just recently updated it, and we'll be updating it again shortly. Access to this site is through www.phosnet.ca, as you can see in the upper corner. If you go to the main Connexus website and scroll to the bottom of the main site, and that's at www.connexus.ca, we're based in Canada, then you can find access to all the websites that I'm talking about today that are currently online. I think what I'd like to do is take you directly to the SigNET website, and I'll do that momentarily. So hopefully you can see on the screen a live feed from the Connexus website, and it's a very user-friendly website. all you do is enter the name of a protein of interest. In this case, I'll type in ERK1 protein kinase. When you type the first 3 characters, it gives you a list of options. And as you go to the site, what you see is a little bit of information about the protein at first, and then at the bottom is a listing of the phosphorylation sites that are known and predicted. In this case, we have about 37 sites that are predicted or known for the ERK1 protein kinase. You'll notice that there's a bunch of buttons under the experiment confirmed column, EXP, CO, and F. And this column has white and green buttons. The white buttons correspond to sites that have been experimentally validated, and if you click on that button, it actually puts the information that's associated with that, with the link, in the information box so you can copy and paste it. But if you take the your cursor and you go over and above the buttons, then the information appears in a little box right next to the data. So for example, the tyrosine 204 site in this TEY sequence is known to be an activating site, and so under the functional column it tells you increases activity, and it tells you when you scroll to the next column that it's phosphorylated by LCK, ERK1, JAK2, and of course MEK1 and MEK2, which are the kinases well known to phosphorylate this site. And it's dephosphorylated by MAP kinase phosphatase 2. Now, to the right of these is another set of 3 columns of buttons. The first gives you a direct link to the PubMed website with information on that. And then the next set of buttons gives you information on the evolutionary conservation of that site. So if we go to that, click that button, you'll see PhosphoNet evolution appear. And PhosphoNet evolution will list for 20 different species the location of that phosphorylation site and the actual number and UniProt or NCBI reference numbers for that site. And if you scroll down a little further, for the different species, it tells you the percent amino acid identity or similarity calculated for the whole cognate protein in the different species relative to the human protein, and the 15 amino acids surrounding the phosphorylation site itself for identity and similarity. Another useful feature of this website is, let me just go back to my previous site. Sorry. Let's see. Here we go. Is the last set of buttons. Now this is the kinase predictor website, and for this website, if we click on it, we developed an algorithm trained with about 22,000 kinase substrate pairs with the phosphorylation sites, and using information on the primary sequence of protein kinases, we then developed the algorithm with the training to take and develop a matrix for the 500 of the human protein kinases. And then for the 1 million human phosphorylation sites, what we did was we tested and scored each of those phosphorylation sites against each of these kinases. And the top 50 kinases are listed. And the highest score that's possible is 1,000. And what you see here is that our algorithm predicts MEK1 and MEK2 as the most likely kinases that phosphorylate that site. We also developed a second analysis that takes into account neighboring phosphorylation sites that might improve the prospect that particular phosphorylation site would be phosphorylated by those kinases. So it's a second way of improving the prediction of the protein kinases. Okay, so if we go back now to the slide presentations, I'm going to go now and discuss more about cancer itself and come back historically for its role in oncology. Obviously, protein kinases were identified early in cancer research as playing a critical role. And of course, the first oncogene discovered indirectly by Peyton Roos more than 100 years ago in the Rouse sarcoma virus is a gene that encodes a protein kinase. And he, of course, received the Nobel Prize for that later in the 60s. Now, the gene that was in the Rous sarcoma virus that was transduced is the SARC gene for sarcoma. Contains three well-known domains. The SH1 domain is the protein kinase domain. The SH2 and SH3 domains are involved in protein interactions. The SH3 domains recognize phosphotyrosine, and SH3 domains recognize regions with rich in polyproline sequences. And of course, a lot of excitement happened when these genes were first sequenced by Mike Bishop and Harold Varmus, and they determined that this was in fact a protein kinase because the SH1 domain was clearly recognized as having homology with cyclic AMP-dependent protein kinase and cyclic GMP-dependent protein kinase from studies from Ed Krebs's lab. What made it particularly intriguing was that that kinase catalyzed the phosphorylation on tyrosine residues, and this was discovered in Tony Hunter's lab shortly thereafter. And the rest is sort of a bit history. Now, as it turns out, many different retroviruses transduce normal cellular genes that may become mutated as part of that process, but they're carried by the virus. And a lot of these genes turn out to encode protein kinases, often protein tyrosine kinases. There are, of course, other proteins that are encoded, including G proteins. And we also know that the virus integration can be close to the site of another normal cellular gene and often result in its overexpression. And many of those genes that are affected encode protein kinases. By the way, most of these tables that are on here, they're very complex, and they're provided for your reference. You can download this material directly from this website, and hopefully it'll be useful in your research and teaching. Now, many of the oncogenes that were identified in the birds and mammals were also found in association with human cancers, as developed by looking to see what genes are amplified in regions that are frequently amplified in cancers. They were also identified by studies of DNA transfection from tumors using Robert Weinberg's approach. So, as it turns out, a lot of these protein tyrosine kinases are receptor-based tyrosine kinases. There's about 60 of them, and they all share this catalytic domain of about 247 or so amino acids in size, and they catalyze the phosphorylation proteins on tyrosine. And many of these mutations that occur in human cancers involve chromosomal rearrangements and formation of chimeric genes, where the protein kinase catalytic domains of receptor tyrosine kinases are often fused with other proteins that in fact induce the dimerization of those proteins so that they may be constitutively active as protein tyrosine kinases. Normally, when we look at these are receptors for growth factors and mitogens, and these induce the dimerization and the activation of these protein kinases with autophosphorylation. Auto in the sense that one catalytic domain is phosphorylating the other chain that's in the dimeric complex. And these set up sites of phosphorylation of proteins. And these kind of translocations of genes that induce constitutive type dimerizations are extremely common in cancers as well. And I have a list here of some of those gene translocations. Now, defining an oncogene, I think, is something that isn't exactly clear. The way I define it as a protein that has a gain of function as a consequence of a mutation, and most of these mutations tend to be very restricted in what they do. They may mimic an activating site, as we see with the BRAF protein, the valine 600E mutation, where the glutamic acid is mimicking the negative charge of a phosphate. And this is in the activation loop of the kinase, as we'll see shortly. It can also result often in the inactivation of an inhibitory region on the protein, as is evident in the KRAS protein. It may be the production of a gene that as a gene product that actually has a gain of function. It catalyzes a reaction that it doesn't normally catalyze. In the case of isocitrate dehydrogenase 1. It may also inhibit the degradation of the protein, so it sticks around longer. And so when we take a look at oncoproteins, we see that they do have these very narrow mutations, and it's generally tends to be point mutations. Very few complex insertions and mutations or deletions are evident. And here, if we take a look at the cosmic database, as an example of this, here we look at KRAS. We can see in the upper panel the location of, across the gene sequence of the KRAS gene, the occurrence of mutations, and these are point mutations. And then below we have deletions that are in blue and red insertions and other complex mutations. So you can see that the pattern is very restricted. And so using this kind of approach, you can take a look at the Cosmic Database and other websites that feature mutation information and start to anticipate whether or not this is in fact potentially an oncoprotein. This is something that we've looked into at Kinexus, and we're making available to you with the OncoNet website I'll be describing to you shortly. Again, in this case, the positioning of a glycine residue mutated to any other amino acid interferes with the binding of the phosphates in GTP and the subsequent hydrolysis of the GTP by the GTPase activity of the RAS protein, and so it stays constitutively active in the GTP-bound form. So when we look at many of these sites in protein kinases, there's a region called the subdomain 7 region that contains the sequence DFG that's highly conserved in about 492 of the human protein kinases. And we also have the subdomain 8 region, which has an APE sequence typically, and intervening between these two sequences, the DFG and the APE, is what we call the activation loop of protein kinases. And this is the location of many of the sites that are known to be activating in protein kinases. We have other work going on at Kinexus that we'll be presenting shortly that shows that, in fact, there's also an auto-inhibition sites closer to the APE region that occurs especially in serine-threonine protein kinases, but that's a story for another day. So in the case of the RAF-B protein, as I mentioned earlier, the valine 600E mutation occurs on the valine, is converted to a glutamic acid, and this results in a mimicking of phosphorylation and hence the constitutive activation of the protein. Okay, so the problem is that with the sequencing of all these genomes, there's about 60 million SNPs that are anticipated to exist. And an SNP is defined simply as a gene where there's a mutation at that location in about 1% of the human population. So in fact, there may be even a lot higher number of these polymorphisms. And the sequencing, the whole sequencing of genomes from tumors in comparison to the data with normal tissues from patients is uncovering shared mutations in genes that encode both proteins and microRNA. And there's been a great deal of interest in identifying these. And we put a lot of effort towards this at Kinexus as well. And there seems to be about 2,500 genes that are abnormally mutated in very diverse types of cancer. And the Cosmic Database from the Sanger Institute, basically with this link, is a great place to look and peruse the gene of interest that you may have about what mutations it may feature in a wide range of different human cancers. Now, with SNP chips, these microarrays allow the identification in patient samples of important SNPs, and that's largely how a lot of the companies that are currently offering services for gene profiling are working, is with these chips to ascertain what people's risks are for different types of cancer and other diseases. I think this is actually quite premature, as I'll explain further in the presentation. And pretty soon, the whole idea of using SNP chips will be supplanted really by genome-wide sequencing as the costs come down. And there are plans to sequence over 100,000 genomes in the UK and other countries in cancer to try to identify oncogenes. Personally, I think we have enough information to already start to identify key players in this disease, or really set of diseases, and I think that's actually a wasted effort at this point in time. Already, we can now identify, and the data here is based on sometimes as many as 100,000 tumors analyzed for mutations. What are the most frequently mutated genes in human cancers? And for those of you that can't see it on the screen, at the top of the list is KRAS, and then NMP1, JAK2, a protein kinase, NRAS, IDH1, isotoxicitic dehydrogenase 1, the protein kinase BRAF, MyD88, FLT3, KT, FGFR3, EGFR, HRAS. And so as you go down the list, what becomes quite evident is the most commonly mutated genes tend to be encoding protein kinases and G proteins in most human cancers. And KRAS is highly mutated. And when you're talking about mutation rate, because these proteins are of different sizes, it actually is much more germane to express them in terms of the mutation rate per 100 amino acids. And so if we do this, we see that in all of these different tumors, about 11.3% of all of these cancers had a mutation in the KRAS protein. And then it drops off quite drastically after that in terms of the mutation rate. So for most of the common genes that are related to cancer that are known oncogenes, they have less than about a 1 to 2% and lower rate of mutation per 100 amino acids. Well, the tumor suppressor genes, they have a quite different profile. And they're typically mutated broadly. They feature many types of mutations, as the general loss of function of these proteins drives cancer development, and it's easy to make something not work by mutating it. So therefore, the entire protein is actually open to mutations that might result in a loss of function. And it could inactivate a catalytic domain. It might prevent the binding of activators to downstream targets. It may stimulate the degradation of a protein. And these tend to be more hereditary type mutations that are occurring. If we take a look at P53, a classical case of a oncoprotein, and it was actually originally identified as a protein that was increased in expression in cancer and thought maybe to be stimulate cancer. In fact, of course, we now know that it's the loss of function of this protein that is driving cancer, and the cells are trying to produce the protein, an ineffectual protein, in order to compensate for the growth signals that are inappropriate for the growth of the cancer. And here we can see that it's loaded with point mutations, complex mutations, insertions, deletions. And so this is a more characteristic pattern for a tumor suppressor protein. Now, we've gone and we've analyzed in our databases what are the most commonly mutated tumor suppressor proteins with these complex patterns. And many of them, as I said, are inherited, and they encode often transcription factors and inhibitors of protein kinases, and also sometimes protein kinases themselves, but largely transcription factors seems to really pop up the most. And of course, P53 and RB. So cyclin-dependent kinase inhibitor, CDKN2A, is actually the most commonly mutated gene in human cancers, and then P53 after that, and the VHL and involved in protein synthesis and P10 follow that. So this gives you an idea of some of the genes, and when we look at the mutation rates, though, they do fall quite dramatically after the first few again. So when we consider the most common forms of human cancer, if there may well be over several hundred oncogenes, and there may be several hundred tumor suppressor genes, and it's combinations of the loss of function of the oncogenes and, sorry, the gain of function of the oncoproteins and the loss of function of the tumor suppressor proteins. The number of combinations can literally be astronomical, could be in the billions. So that's why it's very likely that every person has a very unique form of cancer, although there are certainly certain genes that are the most commonly affected in many cancers. But from an individual basis, you have to figure out for that person what it is. Now, one of the things that we've done at Kinexus is to then analyze for these mutation rates in individual types of solid tumors. And we can start to see patterns that certain genes, the ones that are most commonly affected in cancer, the CDKN2A and the P53 and the KRAS and P10 and NRAS, these seem to be very commonly affected in a wide range of different types of human cancers. But then there are other types of cancers where what we see is that there are certain genes that are highly mutated in association with that cancer. So, for example, the IF1-TM1 gene, it's quite a higher rate in cervical cancer, and the IF-ITM3 gene is also very highly affected in cervical cancer. So we observe, this is the second going further down the list, these are all oncoproteins and tumor suppressor proteins, and the rates of different types of cancer show different preferences for mutated genes. But some tissues, like lung and endometrial cancers, sometimes breast, do show high rates of mutation of many different types of genes. Okay, so if we take a look at the 550 most commonly mutated genes in human cancers, the ones that are defined as oncoproteins or tumor suppressor proteins, we find that that accounts for about half of the genes that we see that are mutated in cancer quite significantly. The other half are actually unclassified. And we can see that for the oncoprotein group, the 78 of those, at least 71 of them clearly have a gain of function. Some of them, though, also are known to be tumor suppressor proteins in a different context. Likewise, with the tumor suppressor proteins, about 140 of the 199 clearly have a loss of function, but there's about 46 where the functional effect is unclear, and some that even in the right context could actually promote cancer. If we look at the distribution of these oncoproteins and tumor suppressor proteins in terms of the categories of different types of families of genes that are affected, the transcription factors show up quite highly, about 14% of the most commonly mutated proteins, and then the protein kinases with tyrosine, and then followed by the serine threonine protein kinases show up quite high. Adhesion proteins and scaffolding proteins are another group, and these give you an idea that certainly protein kinases do show up prominently. Well, this data that I've presented to you, it's in fact can be derived from our open access Onconet website. We have a beta site that's available to look at. It's www.onconet.ca. And currently, it's not officially launched, but a beta version, like I say, is online. And it features information on about 3,000 proteins that have been previously linked to cancer one way or the other. And it tries to contain information both on gene expression patterns and also the mutation. Again, much of the data was originally collected from the COSMIC database. But one of the problems from the COSMIC database is that you can approach it if you know the protein, but it's hard to approach it from the standpoint of a different type of, particular type of cancer. So this is one of the things that we've tried to build into our website. And we've classified the proteins as oncoproteins, tumor suppressor proteins, and this new group of proteins I'd like to talk to you about called tumor requiring proteins, TRPs. Now we can go online to the Onconet website briefly, and if we go online, basically there's a number of options in the menu that you can click on the queries of the types, You can pick a protein of interest, a tumor type, or a protein. And let's take a protein, for example. I'll do a protein listing. Let's do a simple one that's well known, like ERBB2. Just first I'll go to the tumor type protein listing. So now a menu appears, and basically you can list the type of tumor protein you're interested in. Let's say here's a tumor suppressor type. Then you can list different types of functional types if you want to narrow it down, or you can just do the whole list, leave it unspecified. And then you can select from different possible outputs that you can vary the menu, but you can just generate a list of a table of the listing of proteins. Now, this list appears pretty fast, and it'll list a lot of these tumor suppressor proteins. Okay, so I was going to do the listing from, let's say, a protein listing again. You list the, if there's a protein of interest, you can go, you have to list the type of analyses that you want, whether it's a gene mutation analysis or an expression analysis. So we can go gene mutation, and we can select then from the cancer type that we want, or unspecified. But let's pick something like breast cancer, and then pick the top 50 most mutated, and it generates a table. And again, much of this data is retrieved from the Cosmic database and some others. Here you can slide across and start to see some of the values. Now, this is a beta version, so we have not yet added our latest database that has the information from at least 7,000 human tumors for each of the genes. So this version hasn't got all the information there. So we're about to, we have the database already created. What's holding up OncoNet being launched is that when we have descriptions of the proteins, presently you can do a protein description. You can type in the name of the protein you're interested in, like ERB2, little extra B there, and retrieve the information on it. And so we have a lot of detailed information on the protein, and if it's used in clinical trials, sometimes if there's information about drugs that can be used on it. And so we're just updating this information for many more of the genes, and then we'll be launching it. So that's the OncoNet website. Okay, back to our presentation slides. Okay. Okay, so now, sorry, let's go to the next slide. So one of the interesting things that came out of this analysis to look for tumor suppressor proteins and oncoproteins was that we could look at the rate of mutation per amino acids, and we found that the vast majority of the 3,000 entries, including a lot of entries that had nothing to do with cancer, had mutation rates that were typically in the order of about 0.025 to 0.5%. And this is where you can see the bulk of these mutations here. So I'm sorry that the data doesn't appear on the scale for what the percent is. But there is a subgroup here that actually have mutation rates that are far lower than the average that's determined here, both the median and the average rate under percent rate per 100 amino acids in this bottom box to the very left. You see that there's there's a median value of 0.08% roughly of all human genes have mutations in cancers. That is, if you have a bystander gene, there is about 0.08% of the time it will have a mutation every 100 amino acids. It's not that much higher for many genes that are known cancer genes. So this is one of the problems with sequencing the genomes of a large number of people, is that you're going to get this background level going up, and so it may be hard to see mutations that may in fact be contributing to the cancer disease. But what's really, really interesting to me is that there is a subgroup that actually has a much lower rate of mutation. So there's about 176 entries that have a rate that's less than 0.025%, and about 85 entries that had a rate that was less than 0.01%. So these genes apparently will not undergo mutation. Now, obviously, they probably do undergo mutation, but in a growing tumor, if a population, a cell, acquires mutations in these proteins, it probably compromises the survival of that cell. And so there's a process where there's a natural selection that, of course, is favoring the retention of oncoproteins and tumor suppressor protein mutations, but is also restricting any mutations on these tumor-requiring proteins. So one of the interesting questions then is, what are these tumor-requiring proteins? They seem to be resistant to mutations, and we believe that they're required for the actions of oncoproteins. So loss of their function may compromise on the ability of oncoproteins to signal in cells to stimulate cell growth and other activities that are necessary for development of metastasis, for example. So these rates of mutation are at least tenfold lower than in most cancer cells. 72 of these proteins that we analyzed in over 7,000 tumors had only either none or only 2 mutations. Sometimes these TRPs are protein kinases. And so here's a list of what we believe are these tumor-requiring proteins that are the least mutated in human cancers. And they often undergo changes in expression in human cancers, most typically an upregulation in human cancers. And amongst this list, interestingly, is ERB-B3. So this is a protein kinase, a receptor tyrosine kinase. It's thought not to be active. It forms complexes with the EGF receptor and ERB-B2 and ERB-B4. And it's thought that it, in fact, allows, through cross-phosphorylation, although it's not active as a tyrosine kinase itself, it will become phospholipid tyrosine residues, and these set up docking sites for SH2 and PTB domain-containing adapter proteins to then form signaling complexes. I suspect that actually it may in fact be important in cancer action, but not as important as some of these other genes, because its mutation rate's a little bit higher as a TRP. Okay, so what we're seeing then is if we have a receptor signaling complex and the binding of these adapter proteins for signaling, these can turn on, for example, from the SOS activation of RAS, the turning on of a cascade of protein kinases that are serine-threonine kinases, which can culminate in the activation of transcription factors. And we can also, through generation of second messengers like some of the phosphatidylinositol phospholipids, generate second messengers that activate protein kinase C and the PDK1-AKT type signaling pathways. When we look at these pathways and all these integrated proteins, when we look to see how many of these are oncoproteins, these are outlined in green now, you can observe that the these all integrate into a common signaling pathways. If we look at tumor suppressor proteins, these are also integrated into the same pathways, usually regulating the activities of these activated proteins for cell growth. The tumor-requiring proteins, the TRPs outlined in yellow here, RISC isoforms and ERK isoforms, these serine threonine kinases act downstream. They may be required. So these oncoproteins turn out to be quite interesting potentially as both drug targets, because they are protein kinases, and also because they don't undergo mutations so easily. They may in fact not, as drug targets, be as susceptible to development of resistance over time. So I think these represent a very important class of potential targets for drug development. Now, one of the other websites that Conexus has developed is the TranscriptAnnette website. Now, the information in the TranscriptAnnette website is available in the Onconet website, but right now TranscriptAnnette is online, and it's a very useful resource to interrogate a large number of genes, not only in the context of cancer, but also just normal gene expression in diverse types of tissues. So you can do different types of query, and if you click on the button which is hover to view options, you get a list of potential queries that you can do. And one of the queries is the cancer gene expression query. And so for this query, what you observe is that you can pick a particular cancer that you're interested, let's say glioblastomas in the brain, and then you can see what genes are the most upregulated or the most downregulated. And so here we can look to see the most upregulated genes. We hit the search button and it generates a table. And all these tables are downloadable as in MS Excel spreadsheet format. And you can peruse through and observe what the gene expression levels appear to be based on mRNA expression data. Now, the creation of this website was really based on pulling the data from about 5,000 gene expression studies that were deposited in the GEO database of the NCBI. And in some cases, there may be an N of only 1 or 2 for the tissues. But in many cases, what we did was we normalized the data across all of the gene chips using about 60 commonly expressed proteins that were highly expressed in all these various tissues, and then normalized the data to those. So while it's not perfect, it is a very nice place to start. And you can get quite a bit of information on comparing different tissues, how related they are to each other in gene expression. You can look to see basically from a tissue standpoint where are the most common genes, where are the most uniquely expressed genes for that tissue. You can look also from a protein standpoint or gene standpoint, where are these genes most highly expressed or most uniquely expressed or most commonly expressed. Okay, coming back again to the slides. As I said, TranscriptAnnette basically is data is pulled from, as it turns out, about 50,000 studies is featured in the GEO database, but we only pulled the data from the normal tissue and human cancer tissue. So we have about 300 cancer cell lines that are covered, about 250 different types of human cancers, about 50 types of human cancers and about 250 types of different human tissues. Okay, and there's about 21,000 genes that are covered. And this represents about 125 million measurements for gene expression. Okay. So it's an open access resource, and I encourage you to use it. And as it turns out, it's very useful for something I'm going to show you shortly, our KnowledgeBank database. Now, looking at all of these genes that are upregulated in human cancers from this database, we observe that about 12% of the upregulated genes are known oncogenes, and about 12% of them are tumor suppressor genes. But in fact, 76% of these genes don't have a direct link to cancer. If we take a look at the most downregulated genes, 136 of those, 13% of those are oncoproteins and 10% are tumor suppressor proteins. And again, 76% are not really linked to cancer at this point. So this is a listing of some of the genes that are the most commonly upregulated in human cancers. And the way this is read is that each little square represents a different tissue, but the tissues are organized by each row, so they're sorted within each row, so that the strongest signals for an increase is black, and then green, and then yellow is where there's almost no changes, and then red is where they actually go down. So in some tissues, some of these genes, where they're highly increased in expression, in other tissues, they may actually be slightly reduced in expression. But these are the most commonly affected genes. And again, we see that ERB3 is in fact one of the most commonly upregulated protein kinases that we could see in cancer. And so my thinking of ERB3 is that it probably truly is a tumor suppressor protein, and that it's upregulated to inactivate activated receptor tyrosine kinases, and it probably represents an ideal target for the development of monoclonal antibodies. In fact, such antibodies are already in the clinical trials and may in fact be a very useful treatment, perhaps better than some of the ERB2 targeting monoclonal antibodies that are presently on the market. Here's a list of the genes that are the most downregulated in human cancers. Again, dark red represents strong downregulation, and yellow is no change, and green slight increases. So the idea here is that, are these biomarkers for human cancers, and do they provide us with insights into the regulation that's going on in human cancers? Well, one of the things that I would suggest is that really in the previous slide that I showed you with respect to the distribution in these cancers, where we see that there's an upregulation of oncogenes in downregulation of oncogenes in human cancers and upregulation of tumor suppressor genes in human cancers, I don't think it's so easy to predict from these patterns of gene expression which ones are actually contributing to the development of the cancer and which are actually responses of the cell to try to correct for the miscommunication from the mutations of these genes to compensate. And so really in cancer, although there's obviously a battle of the immune system to try to fight the growth of the cancer cell going on in an individual with cancer, there's actually a battle going on inside the cancer cell itself to try to compensate for this defective signaling. And when that growth control is lost, then you get basically unregulated cell growth, and that contributes to the growth of the cancer and then also later metastasis. Okay, so since protein kinases show up repeatedly as very important targets for drug development, currently there's over 19 small molecule drugs that are inhibitors of protein kinases that are effectively used for treatment of cancer. There's more than 150 that are in clinical trials, probably more than 500 that are in preclinical trials. As I mentioned earlier, about a third of all R&D in pharmaceutical industry is targeting protein kinases. Protein kinases, while most of the interest has been from the oncology perspective, protein kinases have been linked to over 400 different diseases. So much of these inhibitors that are being developed for cancer may have much wider applications, for example, in neurological disorders, which are thought to be more affected than with protein kinases than perhaps cancer itself. So there's a great deal of promise here, and I predict that certainly within the next 5 to 10 years, I expect about half of all the R&D development will be targeting protein kinases based on the success that's happened so far. So one of the things that we've created that I think is a really helpful resource for the scientific community is DrugKinase. And DrugKinase is basically a human protein kinase drug knowledge base. It features experimental results that we've collected from the literature, from other websites, for 400 kinases with 850 different compounds that are known to inhibit kinases. And what we recorded is the lowest Ki, IC50, or KD value that's been reported for that particular kinase, for that particular drug in a pair. And we use that data, much as we trained our algorithms to recognize phosphorylation sites for recognition by protein kinases based on the amino acid sequence of the kinase. We also use the training data from the kinase drug pairs and the sequence information on the protein kinases to predict the interactions of another 200,000 kinase drug pairs with 500 of the kinases. So this really allows you to take an existing drug and potentially identify off-target kinases that might be affected by that drug. And it has strong links to other websites and provides information on each of these drugs. So in the interest of time, I encourage you to take a look at the DrugKinase website and visit it and see how it's useful to you. Now the final talk presentation is really based on what we're trying to do with these websites to integrate them into databases. And we have a number of other websites that you can look at. If you take a look at the PowerPoint presentation as we're running low on time, you'll be able to see information on data that was generated at Kinexus using our proteomics technologies. And these websites involve data from immunoblotting and from antibody microarrays. And with our antibody microarray Western blotting website, we have about 10,000 immunoblots that are quantified with data generated for 300 antibodies, over 200,000 measurements, and these are online. And over 95% of this data is actually unpublished, but it's open access. The other database is our SigNET database for antibody microarray, and this data is open access as well. It has results from about 2,500 microarrays that we've run with as many as 850 antibodies on chips. And this is searchable, and again, 99% of the data is unpublished. Now, the last thing I want to talk about is there's two mapping initiatives that are currently underway at Kinexus. One is the connections maps. These are mini maps using only experimental data that's target-centric. That is, you have a protein of interest, usually a kinase, and we're interested in the upstream activators and downstream regulators. And with our antibody microarray analyses, we're going to be generating these maps to map the data from that. But these maps will be available to use for teaching and for other purposes. So here's an example of a map for the JAK2 protein kinase. Again, this is the kinase that's one of the most commonly mutated kinases in human cancers. So it lists the name of the protein and the UniProt ID number in the purple area, and then all the phosphorylation sites that are affected on the map. These are targeted green for activatory, red for inhibitory, gray where the functional effect is unclear. And so we would identify those protein kinases that target these sites directly. If it's known, if it's unknown, then it'll be a direct connection. And autophosphorylation. We also are listing protein interactions with the target protein. And at the same time, we have a list of targets that are downstream of that protein kinase. that are known to be either activated or inhibited. So we are generating hundreds of these maps, and they will be open access and available. The other, though, map that we're most interested in is the KnowledgeBank database. This is the most versatile. This is in development. It'll be online, we hope, shortly. It identifies protein-protein interactions, kinase-substrate interactions, and kinase-drug interactions. And it actually integrates the information and provides links to all these SNPs, TranscriptAnnette, DrugKinase websites, as well as Uniprot and other websites. And the way this works is it generates new maps with the associated protein as the hub of the map. It's in the center of a wheel. So we have an interface that you'll be able to customize with respect to what's being shown, whether it's predicted data and experimental, or experimental only, or only kinases, or only functionally affected proteins. So the kind of map that you're going to see is a link where the center of the map is the target, and whether the interaction is upstream or downstream, indicated by the direction of the arrow. And the size of the icon is related to the amount of the protein. And these maps are tissue-specific or cell-specific. So we'll have about 600 different types of queryable maps for a given protein, depending on the tissue that you're looking at. The length of the distance is related to how strong the phosphorylation connection is as predicted. We also have protein-protein interaction maps, largely pulled from information from the string database. And again, the stronger the score for experimental data, the stronger the interaction. And then we have also a drug interaction database with kinases. And what's unique about these maps is that if you click on one of the peripheral components in the map, it becomes the hub of a new map and shows you all its interacting players. So for example, if you saw a drug that's dasatinib was strong effect on ABL here, you can click on dasatinib and it becomes the center of a hub, and you see all the associated proteins. So I apologize that the talk has gone a little over. There's a lot to cover. There's a lot of people that are responsible for allowing me to put this data together and to create these websites, and I'm quite grateful to them. I'm also thankful to the organizers for the opportunity to present this work, and to you for taking the time out of your busy schedules to see what it is that we're doing. I'll take a few questions in the Q&A section, and if there's any questions, I'd be happy to see them. Presently, I don't see any questions, so I guess we can call that the end of the session. And I urge you to try to take a look at these websites and to use them. I think they're very powerful for designing experiments. And we have other websites that are in development as well. One on protein kinases, another one that's a publication database where a lot of the data I'm showing today will be in a publishable format for you to refer to. So again, thank you very much, and good luck with your own research, and goodbye.